The first endogenous hormone compound to be discovered are now referred to as auxins, or IAA, which is short for indole 3 acetic acid. This is their chemical name. And the word auxin comes from the Greek word meaning to increase, because auxins, generally speaking, but not in all cases, are thought to increase growth. And for each of these endogenous hormones, we will state whether or not they are considered to be a growth promoter or a growth inhibitor. And auxin is the one on our list that we will describe as being both. So we'll say auxins are growth promoting, and in certain situations they can also be growth inhibiting. And we'll talk more about this dual nature here in a few minutes. But let's talk about what things in plants, or what growth responses that auxins actually stimulate or promote in plants. And they stimulate the following: cell enlargement, or the expansion of cytoplasm, and therefore the expansion of the volume of the cell. They also stimulate, in conjunction with another plant hormone, cell division, or mitosis. At very low concentrations, they also stimulate root development, or root growth. And again, that is low concentrations. They're responsible for directional growth in what we would call, as we discussed earlier, tropic responses, not tropic, but tropic responses, or growth responses. And as discussed also earlier, they're thought to be the main hormone responsible for the phenomenon of apical dominance. And so this is the big exception, and this is where, in this case, we think of auxin as being a growth inhibitor, because at high concentrations, very high concentrations, they have been shown to actually inhibit the growth of things like subapical meristems. And so there's that dual nature. These we would all consider pretty much positive growth, but again, this would be more of a negative growth factor, and so that's where it gets the promoting or inhibiting label. Now, another thing to keep in mind here is the fact that I say that at low concentrations, root growth is stimulated. The same concentration in leaf tissue may not have any response at all, whereas the concentration of auxins in leaf tissue that cause cell division and cell enlargement, or in other words, growth, in the roots, they can actually inhibit the growth. And so there's another example of a possible inhibitory effect. Auxins are actually used, or synthetic forms of auxins are used commonly to stimulate root growth at very low dilute concentrations in the nursery industry. Plant propagation, they'll take cuttings of a woody plant, dip it in a very low concentration of auxin solution, or a synthetic version of an auxin, and then put it into moist soil in a greenhouse, and those stems, although they didn't have roots before, will begin to, in many cases, sprout roots. Auxins, once again, inhibit subapical shoot growth, like we already said here, and again, root growth at high concentrations. So that's pretty much what auxins do. There are several synthetic, should be a Y, synthetic versions of auxins available, and they're in wide use in agriculture. The most widely used is a chemical called 2,4-D, and 2,4-D is used as a herbicide. And basically, it's a high concentration of auxin, to the point that it stimulates excessive growth. It stimulates the plant to grow uncontrollably, and it starts to grow and divide its cells at a rate that's faster than it can keep up with in terms of uptaking water and nutrients from the soil. And so plants that have 2,4-D applied to them often look very spindly, and they'll start, instead of nice straight growing shoots. The shoots will often just start turning on themselves and be white and bleached looking because the leaves and the stems don't have enough time to develop proper chloroplasts and chlorophyll that would make them their normal green color. One other little note on auxins that I'll talk about here briefly is how they were discovered. And they were discovered by studying how oat seedlings grow. They were looking at oat seedlings, and oat seedlings, when they germinate, they would start growing roots. And in addition to that, they would grow shoots. And they noticed that when they had a light source, the sun coming through a window or maybe a light fixture in an indoor room, these shoots of the oat seedling, let's say the light source is over here, there's my little sun, the seedlings would curve or bend their growth directionally towards the light. And again, that's what we would call phototropism. They proposed that there must be some sort of signal coming from the tip of these shoots to the tissue behind it that would cause, let's say, this region, use red here, of cells to elongate more rapidly than the cells on this side of the shoot, causing that bending of the shoot growth towards the light source. And so they did a number of experiments, and they looked at the actual tips themselves, and they'd come in and cut the tips clean off of the shoot and take that tip, and they'd set it on top of a gel-like material called agar and let it sit there for a while. Then they could get rid of or throw away the shoot tip and take that piece of agar and put it back on the shoot in place of the tip. Now, if they put a piece all the way across the tip, well, then it would just grow straight ahead. But if they took it and, well, wrong color here, but you get the idea. If they put it to the one side of the shoot, well, then that growth would curve because those cells would divide on this side more rapidly or elongate at a greater rate than they would on this side of the shoot. And so that pretty much proved that there existed some sort of chemical signal that was being produced by these tips and being transported back to the tissue behind it. And this is how they discovered auxins.